Okay, so today we will be talking about manipulating neural activity, which is the ultimate, I think, the most important part of today's this semester. So let's see. So our goal for today's course is to develop an experimental logic behind the gain of function and loss of function experiments. Particularly gain of function is to discuss about a sufficiency in some kind of a behavior, and loss of function is also another necessity criterion, which we will go through a little more. And for the nervous system, we will be covering various approaches for manipulating neural activity of various organisms, including humans. So the picture on the bottom is actually depicting on kind of emerging, it's already emerged on optogenetics and comparison with the electrical stimulation. So you see electrical stimulation can do not discriminate different cell types, but it affects the local cells, neurons, while optogenetics has an excitation and inhibition, which is only affecting those affected cells. So we will be discussing this further. So given many neural activity manipulation techniques, I will be trying to covering these black ones, but cannot cover whole things. So those who are more interested, there's another class, lectures in the graduate school. So first I will be talking about physical manipulation, which involves an ablation or a thermal control. And second, we'll be using electricity, so electrical manipulation by use of microelectrode to stimulate, or even we can use excessive current to lysis the tissue. It's called electrolytic lesions. And another is a pharmacological manipulation by using drugs. Okay, so drugs, there are names of responding to the receptor and agonist, antagonist, and also delivering how the delivered drug, especially the central nervous system. And we also will talk about genetic manipulation, which is the perturbing endogenous genes. And we actually can have a specificity in terms of our target by expressing genes to particular types of neurons. And pharmacogenetics, which is also called as a chemogenetics, is we won't be getting deep into this, but I'll just tell you that by using transgenes from other species, we can have a designer receptor, which is activated by designer drugs called DRAGs. And this is a little bit beyond of the scope, but I'm telling you, like optogenetics, we can have a specific cell types of neurons by giving a drug to activate those. Lastly, optogenetics is an emerging technique which is quite exciting and which is shown in the world in 2005. Probably you guys are already born by the time. So this is by using channel rhodopsin 2 and halo rhodopsin and archirhodopsin. And what does it do is it uses a life's intrinsic creation of light-sensitive ion channels or pumps. We use it to engineer this into the neuron, and we use light to control the activity of neurons. So this is so cool technique which revolutionized the world of neuroscience. So we will be discussing more. And neuromodulation in humans will be lightly touching, but this is kind of the very hot area of research that we can use of various kinds of these technologies into translate into the human treatment of neurological diseases. So pharmacology, microstimulation, and so-called TMS or transcranial magnetic stimulation or even we can use ultrasound for neuromodulation. These are something emerging one in the future. I think we'll see more and more to treat human diseases, because so far many pharmacological approaches has been limited. So we are considering a new era of digital drug, or like these neuromodulation, which may be a non-pharmacological way. So we will be expecting a lot to see more of this in the future. Okay, then the first question to ask ourselves is, why do we want to manipulate neural activity? That sounds like artificial, okay? The reason for this is at the end of our lecture series is the ability to engineer and manipulate means we have a lot of control over the system. When we try to understand, let's say a cell phone here, cell phone, the best way is we give input and measure the output, changing the input and observe what changes as an output. So this is kind of engineering way of understanding a system. Even if the system is in a pathologic condition, in disease state, we can even try to treat this by understanding the inner workings of the system, like reverse engineering. All right, in that regards, perturbing a neural system to test our hypothesis about the role of the neural system. In neuroscience, especially, that could be mental processes and behaviors, which is related to our health and disease. Okay, let's just have a thought experiment. So let's say I'm interested in appetite regulation, okay? So we have a target location, let's say in a potential target location in some of the brain area. So we can have a hypothesis that maybe a population of neurons, say this area is regulating appetite. How would you design your experiment and to determine the necessity or sufficiency for food seeking? So this is the most important aspect when we consider about manipulating neural activity. We want to explore, let's say, a particular area of the brain. So we have set up a hypothesis. So is this area required for appetite? So maybe you can actually just eliminate that part of the brain, took it out, and then see if probably it's an animal. See if the animal is not eating anymore. Then we can at least think, oh, maybe the role of that area isn't necessary for food seeking, okay? So, but you have to be very careful about interpretation of this. Let's say I took out, let's say this POMC area, I eliminate by ablation, and then the animal is not seeking food anymore. But you can say this is actually appetite gene or appetite neuron you discover. That's probably, you gotta be careful, it's wrong. Because with your assay of food seeking, when you take this one out, the animal may be sick so that she cannot eat anymore. Or the animal, this area may be affecting the motor activity, and the animal just cannot move to actively eat food. So you gotta be very careful on interpretation. So your design of this, it's only this area would be at least necessary for food seeking. Or what about this area? You want to, on the other hand, like stimulate the area to activate that neurons in that particular population. And you realize the animal is now start eating, keep seeking the food. Oh, then you can at least see this reason of neural activity is sufficient to cause the animal to having more food taking. So this is an example of why manipulating neural activity is so important to find out a causal relationship, especially this complicated neural system. You want to correlate neurons, distribution of a neuron population and behavior. You want to find out the causal relation. This is very important to be able to manipulate neural activity. And this example is a new brain circuit in feeding control, which came out 2 years ago in Science. Okay, so then let's talk about methods of manipulating neural activity. So this simple cartoon from the textbook, I think explains a lot. So first, it shows a physical manipulation. It shows a blade. So, you know, it's like a neurosurgeon who are, let's say, removing part of brain. If you find out an epilepsy patient, persistent epilepsy, no drug works, you know what the neurosurgeon do? Identify the focus of the epilepsy start, and you just using blade and cut it out of the brain, okay? Potentially this can have a lot of side effects, but you know, definitely this is a physical way. Another example is in electricity. So, you know, still now a severe depression, which is resistant to any drugs, you know, the patient can undergo electroconvulsive therapy, which is, you know, you give a shock to the brain, okay, and then reset the brain, which sometimes, you know, reducing the severe depression. So electrical definitely affects a good way for manipulating. However you think about it, electricity is about, you know, current flow in the brain tissue. And so it's a little hard to localize and hard to prevent this effect from, you know, the path of this, not only neural cells, but also axonal tract. So this has a little bit non-specific. On the other hand, you can also consider just sending a drug into the brain directly. So it's pharmacological, either IV or direct injection or intake. So it's a pharmacological control or manipulation neural activity is quite effective. In fact, many drugs, modern drugs are using pharmacology. But for manipulating neural activity, you have to inject locally. You see the diffusion of the drug, so the localization may be affected. And also, you have to consider about the pharmacokinetics and pharmacodynamics of this interaction. So it might not be very easy to make a precise temporal control. On the other hand, the lower part, you'll see genetic, genetic, genetic. And you'll see some neurons are labeled in the dark black outline. What this depicts is now genetic modification, you can select specific cell types, okay? So you now achieve specificity by using a genetic approach. So you can label specific cell types which can respond. So in this case, you can selectively delete or kill the cell types of, in this case, a green cell. In this pharmacogenetic case, you give a drug which will only affect these genetically labeled ones. On the other hand, similarly, by using optogenetics, you can use light to only elicit neural activity change to these specific cell types. Here I just put the CNO is for pharmacogenetics or chemogenetics, a drug used for this, which is a precursor for clozapine in vivo for chemogenetics. Okay, so now let's discuss about experimental logic. So let's say we have an animal model. We are interested in a specific brain area. So let's say in this case, let me give you an example from last year's Science, which is a very cool technique. But this is optogenetics. So you shine the light to local area of the brain, and you find the animal jumps, okay? Then this stimulated region is involved in the jumping behavior. So Julie, can you guess what kind of experiment of this? Is it gain of function or loss of function?
I think it's gain of function.
Exactly. Very good point. Okay. On the other hand, let's say you pick an area of the mouse brain location, and you actually lesion that space, the place. So either ablation, and you realize the mouse now forgets spatial memory, so it can't find the location very well. Then the lesioned region was at least necessary for recall of the memory. So let's see, Hanjinu, can you guess what kind of study is this?
Lots of
Yes, exactly. So by losing that function, you actually find out the necessity. And by stimulating, you find the sufficiency of this certain behavior. So this is a basic logic why manipulating neural activities is so important and crucial. And this example, I just show you a cool idea that, like let's say VTA area is quite deep inside the brain, and it's very challenging for optogenetic to shine light deep inside. So a clever technique here is a UCNP called up-converting nanoparticle, which makes a near-infrared light. It converts into half of the wavelength, so it make it blue. And some of channel adoption requires a blue light so that near-infrared can go deep inside the brain. They can excite up-converting nanoparticle. They generate blue light, and that will excite a specific neuron of our interest, which is deep inside. So it's cool, and this is why it's in science. All right, so now let's go back methods of manipulating neural activity. So physical method is basically ablate neural tissue. Another physical mechanical method is a thermal cooling, okay? So which is you can control the temperature of the nervous system, and you can control neural activity. Especially when you cool down, everything slows down. So that suppresses neural activity. However, you have to remember that our brain or animal brain has a very tight control of thermal regulation, okay? Because temperature is very important for all this biochemical activity and enzyme activity. But we can actually use this. Another electrical activity is applying current to a tissue, either to stimulate or cause a lesion. So one cool way is you use electrical stimulation, and then at the end of the study, you can actually cause a lesion by electrocautery, so that later on you can actually check by histology. I'll show you the example. Pharmacological methods using chemical compounds or drugs to alter the function of ion channels and cell receptors. And genetically encoded transgenes will be used to ablate neurons or change the baseline physiological firing properties. Lastly, optogenetic methods is targeting those light-sensitive ion channels and proteins to specific chosen neurons for our neuromodulation. So optogenetic methods is a kind of a revolution, and we will be looking into a little more detail. So let's go look at one by one. Physical manipulation. So ablation is basically removing tissue by surgery or melting or evaporation. So that's ablation. So we create a brain lesion, okay, by exciting or aspirating, okay, or by suction you can do it during a stereotactic surgery. Why do we want to do this ablation? We want to study the necessity of a particular brain region for a biological function. Okay, so this is a really crude method, right? Performing, basically you took out the area the reason. So that's a loss of function experiment in the brain for the necessity. So you can easily consider what are the limitations, okay? It's basically you can't get it back, so it's irreversible. And often difficult to control the precision of the lesion. You know, when you do surgery, human brain may be big. If it's an animal, it's small, your hand shaking, and the precision while you're observing, probably using a stereotactic microscope, still there are issues for precision, okay? And also you have to think, hey, I want to remove, let's say, hippocampus of a rat to study memory, but the act of cutting it out inevitably cuts the passing through white matter axonal tract, which can have a significant systemic effect, which is hard to control, right? So we don't know the impact of removing tissue on the remaining brain structure if they are all connected, okay? Neuron or axonal fibers. So obviously this is a crude method, but it can, it can be useful. So, but I want to show an like advanced technique for ablation because that's crude. Why don't we use like microscopic ablation, which is using focusing the light, which can create a femtoliter tiny volume, and you have the energy. So you can even cut single axon track, okay? And then you can observe over time how they reconnect and regenerate. Well, this is a cool example that it's possible that we can do ablation very precisely. It's called laser axotomy, meaning that axon and thumb means cut, so you can cut a single axon by using like femtosecond laser, which is typically used for 2-photon microscope. Okay, so you can do very cool experiment with this. And the second physical manipulation I'm talking about, thermal cooling. So what does a cooling mean? So those who you remember primary cell culture, you always use a cold ice and make the cell cold in the middle because it will slow down the kinetics of various biochemical events, such as ion channels and the cell physiology. So if you do it very mild and in a limited range, then as far as you alter temperature will not cause long-lasting damage. So that's what you can control. So this experiment is at least reversible. Of course, you do it too much, you like freeze the cell, maybe part of the cell will die, but so basically it's reversible too. But the limitation we have to consider is the cooling is based on heat transfer method, so that may not be spatially and temporally very precise. The reason is spatially, you probably use various probe. The physical size might be not that small. Also, the process of heat diffusion requires time. It's not like it may not be very fast. So that has a limit. And it may not be suitable for this experiment. So loss of function may be good, but gain of function is another thing to consider because you can heat up so that it may increase in neural activity, but the problem is it can cause damage often if you increase the temperature too high. So this may not be good for gain of function experiment. Okay, so I bring an example of this very cool technique, which came out on Nature in 2008. So, songbird is a very good experimental model for studying speech, okay? So why this study is came out in Nature is the experimenter actually uses a Peltier cooling device to cool down a part of the bird's brain called HVC, which is a high vocal center, and RA, robust nucleus of the archopelium. So these are very important part. For deep inside, you see it's a probe is going into deep, and they make a device to cool down and see the behavioral change, which is this diagram. X-axis is a time, y-axis is a sound spectrum. Okay, so the control is there. You warm the place, you see it shortens, which means the activity gets faster. And you cool down, cool down, cool down, and you realize something's elongated. And you're curious about to compare to control. So you elongate the control and you realize it's almost the same structure. So cooling down, and you find this a very precise elongation of the behavior. So this gives you by first demonstration of temperature to analyze temporal dynamics in songbird motor pathway. So this is a very interesting example of how heating or thermal control can adjust neuronal behavior. So, and let's talk about electrical manipulation. I would say currently the most relevant for human neuromodulation is still using electricity, okay? So especially using microelectrode to deliver electrical current in the brain. Not only recording electrical activity, this is cool. You use electrical recording and also use electrode to actually send currents to stimulate. So it's called microstimulation because typically you use microelectrodes. But you also have to be a little careful that too much current can kill the nearby cells and create, kill here is meaning like, melting away. So that's called electrolytic reason, permanently ablate the neurons, right? So what's good about this? The good about this is, our body itself, our tissues are all immersed in aqueous. So our body is conducting, okay, conductor. So you can basically use electricity, and good thing is by using electricity, we have a good temporal resolution, and we can even use different frequencies. So frequency and different timing can be very, very precise. You can do either reversible electrical stimulation or irreversible by too much current, just making electrolysis. Okay, what are the limitations? Because it's all like conductor, the location focus might be hard. So poor spatial resolution would be still a limitation for electrical manipulation. Okay, so I'll give you an example for that. There's an area in the brain called infralimbic cortex, which is known to be related to a fear. So I show you over here, infralimbic cortex of a rat. So you see this a nice focal lesion, okay? And that has came out in Nature in 2002. So at the area is a medial prefrontal cortex is related to fear extinction. Okay, what this experiment is about. So infralimbic stimulation together with a tone, it's a sound related to freeze. So let me explain you a little bit of this cool experiment. So the rats are in the cage. You remember Pavlov experiment. You give a sound, and then afterwards, let's say 5 seconds later, you give an electric shock. Then the animal now associate the sound before the shock, okay? The next time you hear the sound, you freeze because you remember that event. So the conditioning you see freezing will be 100%, going very high on the day 1. And then what happens on day 2, they still remember. So you give a tone and they will freeze, freeze, freeze. There is a little decrease. This decrease of freeze without shock is called extinction. It's related to memory. But what surprises here is they find infralimbic cortex to use electrical stimulation and they forget already. So they don't freeze the next day. And even day 3, they don't freeze. You see, this is one simple data, but it's Nature paper. What it means is clearly show this area is related to this extinction of fear memory. All right, so brief infralimbic stimulation paired with tones simulate extinction memory. So an example for electrolysis. So I just give you, after the experiment, they actually give a high current to kill the tissue so that after histology, you can see where the electrode was precisely located or not. So this is an experimental technique. And we still, I said, use a lot electrical for human neuromodulation. For example, Parkinsonian patients have this deep brain stimulator, okay, in deep inside the brain, and motor cortex stimulator is here. It requires a certain surgical procedure, and non-invasive way is a transcranial magnetic stimulation, which we use it a lot without knowing a full mechanism still, okay? And also vagus neurostimulator, and some people with pain can use spinal cord stimulator. Okay, this the field of this is still like in its infancy, that we do not know what happens, why it happens, but there's a huge demand of this neuromodulation. All right, so next, pharmacological manipulation. I changed the slide order to make it easier for you. So first of all, many targets of the drugs is actually related to the cell membrane bound receptors, okay? So binding of a ligand causes this receptor activation, which will send out intracellular signaling pathway, which eventually changes in cell physiology. So how to perturb this? So there's a way called the agonist, okay? So agonist binds to a receptor which causes receptor activation. So that is called agonist. And one example in neuroscience is a GABA receptor called a inhibitory neurotransmitter GABA. So this, for the drug, for this is mucimol. And the opposite way is it's called antagonist, which is actually this yellow one. Yellow one is the original one. Antagonists you give, they will bind instead of this original ligand and blocks the activation of this. So binding of antagonist to a receptor do not cause activation. So it blocks the ligand from binding, so that it blocks the normal functioning. So a good example for us is actually sodium, voltage-gated sodium channel blocker, okay? That's called tetrodotoxin, TTX, you will see a lot. This is originally coming from puffish toxin, okay? And the other is, yeah, voltage-gated potassium channel, which is called tetraethyl ammonium. So there are these specific drugs which you can actually utilize for your experiment in pharmacological way. So influencing neural activity pharmacological using chemical compound, which is selectively binds to membrane-bound receptors. So, you know, these are usually reversible, okay? But temporarily, it's not very precise. You can have like millisecond, second resolution, but more or less like minutes or tens of minutes or hours. So agonist can bind to and cause activation of a receptor, okay, that mimics endogenous ligand or a neurotransmitter. So one example is morphine, which mimics the action of endorphin. Endorphin is our endogenous ones, but morphine is a drug, okay? Antagonist binds to receptor, but it will not cause activation, preventing biological activity of the endogenous ligand because it blocks that endogenous ligand to bind. So tetrodotoxin to inhibit eventually action potential, okay? Because it targets voltage-gated sodium channel. So it's toxin, you know, like if you got this toxin, probably it'll be very dangerous. Okay, so how do we give the drug to a particular area in our central nervous system? For peripheral nervous system, it may be easier But central nervous system, especially brain, there's called a BBB or blood-brain barrier. So it may not be very easy to deliver drugs because our brain actively blocks some other chemicals entering into the brain tissue. So a good way, a problem is delivery of drug in vivo. So I give you two examples of first IP, which is intraperitoneal injection, which actually eventually drains through into the vena cava so that it actually mixes with the circulation, okay? However, maybe inside the brain, this can have a problem. It can have a systemic effect. So inside the brain tissue, you have to think another way. So that is by using CSF, cerebrospinal fluid. So if you access to directly that by using intracerebral ventricular injection, such as this. So you have a stereotactic procedure to target that ventricle. And this ventricle contains CSF, which directly meets with all these cells in the brain. So you can, later you can actually check with this dye, lateral ventricle, IV of ventricle, you can do this delivery. So one example is for those moms who have a delivery, have a anesthesia called a epidural. So that is actually injecting drug into the spinal, cerebrospinal fluid area. So that directly affects our central nervous system. So this is a bioprotocol which may be helpful for you if you want to do this. But the important thing I want to point out is you have this area, but you have on the other end, another one is called dummy cannula. So this will serve as a control, okay? And another example here for this drug delivery, you need to have a precise pump, and Hamilton syringe is a very small little amount can be delivered precisely and using a program of micro pumps. And you can use air bubble to see how this is moving. You can have a vehicle or drug, and then you can do this intracerebral ventricular injection. Okay, so this is more practical aspect when you actually do. So another one which I didn't show you here is called the intrathecal injection, which directly target the backside of this ventricle, and then you can actually deliver drugs directly affecting brain, or you can also put this one into the spinal cord of the animal so that you can actually paralyze and you can test the anesthesia or the effect of the drug. All right, now we'll talk about genetic manipulation. So genetic modulation can target specific cell types in the brain using endogenous promoters and other genetic regulatory elements, which will limit expression of genes to specific population of neurons. So with this, you can permanently ablate a specific cell type. An example is ataxia transgene can kill specific neurons, okay? And you can modulate neural activity by overexpressing or knockout ion channel or membrane-bound receptors, which will regulate cellular excitability. You know how action potential works, but these ion channel overexpression and knockout will modulate neural activity. And recently, targeting genetically encoded actuators to neurons can reversely stimulate or inhibit neural activity with high temporal precision. So this is a revolution, and why this is possible. A good example is optogenetics. So you can consider the neuroscience big changes as a optogenetics before and after. This first paper came out in 2005, Nature Neuroscience, but there were people who actually asked for this. So this is a reference for the reasoning, physical, electrical, pharmacological, genetic tools, and the precision. So you can go through later on with this. And I want to introduce you guys already know about him, Francis Crick. which is known for discovering the structure of DNA. But he has done a lot other, especially in neuroscience as well. So I want to quote what he said. The major challenges facing neuroscience, in 1979, before many of you guys were born, he called for a technology by which all neurons of just one type can be controlled, leaving the other neurons more or less unaltered. That's what he imagined. It'll be good to have this. But you know what? After decades and decades, after over 2 decades later, the technology has come, and that's called optogenetics. So optogenetic manipulation, which revolutionized the field. Why? Because you can activate the neuron or inhibit the neuron by adapting a membrane-bound ion channels, which is in fact found in opsin, microbial opsin, okay? And then you can use, by controlling light shining different color, you can actually now control neural activity. That's very, very good. How do we do that? Because this one requires a genetic modification, so-called the opsin gene, and you have to select cell type so you have a promoter region. And how to deliver this into the neuron of your interest? You use a virus as carrier. And then those specific neurons are expressing this specific opsin gene, then you actually shine the light. You need a technology for how to deliver light, such as using optical fibers. Okay. So optogenetics is a set of neuromodulation tools that are activated by light. There comes an opto and are genetically encoded. There comes genetics. So this optogenetic transgenes can be delivered with high spatial precision by using stereotactic surgery to specific cell types by using specific promoter of your own interest, okay, in the nervous system. And so this is just, in our history, it's an unprecedented, because now you can control spatial and temporal. Temporal, you use this light, and light can be controlled by electricity. You can have very precise temporal control by delivering light to target neuron. So then what kind of a toolkits are available as a optogenetic transgene? So you engineered version of microbial opsin. So opsin, you remember, like your retina has a rhodopsin, that opsin, okay? So that's a protein that translocate ions across membrane in response to stimulate by specific wavelength of light. In fact, those lights are, turn out to be visible light range, because we all evolved together in this sunny Earth condition. All right, so there are like 3 things I want to introduce. So think about this cytosol. This is extracellular space. And you remember sodium concentration is high outside, okay? And this membrane potential resting is minus 65 or minus 70 millivolt. So when we shine blue light, this is a non-specific, but a lot of sodium ions came in. So it will depolarize. So it'll, generate action potential. So people find another kind of now ion pumps. So halorhodopsin is a chloride pump. Archirhodopsin is hydrogen pump. So you see this one, Cl minus gets inside. So it actually blocks action potential. And archirhodopsin, expels hydrogen atom by pumping. So it also prevents action potential. So this is very cool. Activation and inhibition is possible by just using light. All right, more details are here, that activation for 70 nanometer, and it can increase a lot of sodium ions getting in. There are another different variants. You will have probably studied this in more advanced course. But the point is, by giving light, you see this blue light make a action potential. Now inhibition is a little different. By giving blue, giving action potential, but by giving this yellow light for this halorhodopsin, and you actually block this action potential. So this is a very, very cool technique. So I'll go over like a couple of these ones. Optogenetic stimulation of this channel rhodopsin 2, which is in fact non-specific cation channel. Okay, where does it come from? It's not from human. It's not from vertebrate animals. It's from green algae. Okay, see this algae, you know, they have a sensor to the light, so that we are using it. Upon observing blue light, channel rhodopsin 2 causing cations to pass membrane. I said this is non-specific, so it's actually down the concentration gradient. So by diffusion, resulting in a rapid, as I said, you see these sodium ions getting in, it actually rapidly depolarizes, which can cause action potential. And what's really cool about this is this can be so fast that you can have even 40 hertz activation is possible. But you don't want to, you know, go even higher. It depends on the kinetics of this channel. And yellow light case, you see yellow light by giving dot, 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 dot activation of action potential. You can block this one by shining yellow light with this halorhodopsin, which is a chloride pump. So Cl minus goes in, so you know membrane potential goes down. Okay. Okay, so that's optogenetic inhibition of NPHR and archirhodopsin. What does it do? It hyperpolarize so that it inhibits neural activity. So there are two kinds. Halorhodopsin, which is a chloride pump from halobacteria. Okay, so it pumps chloride ion into the cell with yellow light. And similarly, archirhodopsin is hydrogen ion pump outside of the cell. So it expels hydrogen ion from this species. And you can think it very easy, but you have to consider in terms of optogenetic inhibition case, there are practical issues. Because to block this, you have to continuously deliver yellow light for sustained hyperpolarization, okay? While considering channel rhodopsin, you just give a shot of single blue light, it will cause action potential. But to block, you have to keep this one, okay? Blue light to mimic action potential. So, you know, in the past, people like complained that, yeah, optogenetic inhibition doesn't seem to work as well in practice. And this is the reason. And you have to also concerned about like side effects due to heating and phototoxicity, which may cause neuronal damage. So that's something you have to consider in terms of optogenetic inhibition. And let's go into more practical aspects. Methods of delivering light to the nervous system. How do you deliver the light? Now here comes an engineering, okay? You can implant a guide cannula for placing fiber optic cable to target anywhere in the brain. If it's on the top surface cortex, you can use LED. If it's just some specific location inside the brain, you can use stereotaxy to target that part. Okay, so that's how it does. And let's see an example. So optogenetics in action. I want to share what I did actually. So I went to Stanford, Diceroth lab, and you guys probably remember this person because we think he will get Nobel Prize in a few years. Okay, so in the lab, I actually performed an M2 mouse motor cortex of this this channel rhodopsin mouse. So I give this, you can hear it.
Yeah.
This is an experiment. So it's a left M2, it moves the right foot. Stop.
It fell. Okay.
And then turn on the light, and then it
Stop.
like this.
Oh my gosh.
Stop. Oh, the problem is it was a little too high, so it makes the seizure.
Yeah.
Thank you so much.
So yeah, this is an example that you can actually control the mouse movement, like remote controlling. And this is another experiment I've done in Quebec City. So this is a mouse with the optogenetic stimulation and whisker. So I just want to show.
Yeah.
how
But it keep on. Oh no.
I
So
can
this
just
is
flash it
optogenetic
myself.
stimulation.
Oh my god.
You see the
And
the
I can
tiny
hear
sound?
it too.
That's a neuronal firing.
Oh, yeah,
Brrrr, brrrr.
And also I want to see whether the whisker the
And this is a
piezo
We
movement.
see a lot of the mouse.
which moves the
Not really
whisker,
much.
particularly.
You want to do it without showing the mouse, you mean?
If we don't see too bloody thing, I don't.
Okay, I just want to show some example. So as a result, what we got is having an optogenetic channel rhodopsin stimulation, and we give a whisker stimulation, which depending on neuron type, some inhibit, some actually amplify the neuronal firing. So I just want to give you some example from my own experience. Okay, so what do we use it for? Studying neural circuits using optogenetics. So typical experiments, you have to transduce a population of neurons with an optogenetic transgene. So you need to use a virus, and you need to have a stereotactic surgery to provide these transgenes. And then you want to investigate the effect directly stimulating or inhibiting the neurons with light. If it's deep inside, you will probably need a optical fiber implant. So there are review papers, projection confirmed with optogenetic. So there are many neural circuits now being discovered for its connection. Okay, so that's very, very cool. And I want to have these methods of stimulating neural activity, category, the tools, and invasiveness, and spatial temporal precision. You know, I won't get into one by one, but all of these, you can actually read it later. This will be a good reference. And also inhibiting neural activity. Okay, so as the last one, I want to give you another photo by Sidney Brenner. So this person is a Sidney Brenner, who is a, when I was a second-year graduate student, he got Nobel Prize. And so I watched him come and actually giving a lecture, okay? So, so what he's known for is he uses C. elegans as an animal model because it has very few, like about less than 1,000 neurons, which is all discovered or figured out now. So it becomes a very good tool for neuroscience. For that, he got Nobel Prize in 2002. And this is what it said: progress of science depends on new techniques and new discoveries and new ideas, and probably in that order. So this is very cool, showing how neuroscience technology can contribute to understanding this science. And I was looking for this, and actually it's very hard to find, but I realized, at least I found this origin, that this is the quote. He actually didn't say it, but this was written by Robertson. Sidney Brenner had in mind when he remarked, and this is the part. So it's good. It's like searching this. And here he actually worked with Francis Crick and MRC molecular in Cambridge, and she is a co-worker at that time. All right, let's summarize. In the past decades, a dramatic expansion and the toolkit used to manipulate neural activity in our model organisms. And we have to know their own advantages and disadvantages. As recent development of genetically encoded tools, which can reversely affect neural activity with unprecedented spatial and temporal and cell specificity, which allows to prove many previously intractable questions about the role of specific neural systems and cells and circuits related to behavior and our human neurological disease. So with that, I think I'm gonna finish this semester's course. So let's see. Let me see.